Hello, everyone, and thank you for joining us today for another ACE Spotlight presentation. My name is Rouzbeh Afrasiabi, and I will be your host. Today, we have Shangying Wang, who is a postdoctoral researcher at the Department of Biomedical Engineering, Duke University. Today, she will be talking about her recent work titled Massive Computational Acceleration by Using Neural Networks to Emulate Mechanism-Based Biological Models. With that brief introduction, and without further ado, I pass the mic to Shangying to give her presentation.
Thank you for introduction. Hello, everyone. So today my topic is the title of my paper that's published in Nature Communications. So before I go dive into that, I will talk to you a little bit about the background. There are lots of species in the nature. I'm always amazed about their design and patterns, including human beings. We all grow from single cells. If we are thinking human being is a kind of pattern, how amazed this pattern can all be developed from a single cell. Even for bacteria is a single cell living creature. Some of the bacterial strands can form very beautiful patterns. So today we'll focus on bacterial patterns. And pattern forming process can be controlled by many factors. So I listed a few. It can be controlled by growth rate, lysis rate, diffusion rate, and etc. Pattern forming processes are very complicated, yet very organized, even with the existence of intrinsic and extrinsic noises. How is that possible? There's a very famous Nobel physicist, Richard Feynman. He said, "What I cannot create, I do not understand." So this is often used by synthetic biologists. However, the truth is, even for things we created, we may still not fully understand. Then this is a very brief introduction about synthetic systems. So in nature systems, there are lots of elements and pathways. The key components that are responsible for the forming of a pattern are always buried in a complex network. It is very challenging to uncover the mechanism underlying the pattern formation only on the basis of studies in nature systems, because one perturbation can cause changes of lots of elements. For synthetic systems, if we can design a gene circuits, then if this circuit can produce a pattern, then we're sure this pattern is formed by these circuits because it's relatively insulated. However, there is still a bottleneck because designing these synthetic circuits remains a big challenge due to the underlying theoretical complexity. So this is an example of a self-organized pattern formation in engineered bacteria. So this circuit was developed by our lab. So far, there's very little researches on developing the self-organized pattern formation in living bacteria because it's very difficult. I'll talk briefly about these circuits. So first, gene T7 will produce a T7 protein. This T7 protein can behave like an RNA polymerase to promote the gene expression of LUX-R, LUX-I, and it can also promote expression of itself gene expression. The LUX-R will produce R protein, and this LUX-I will produce these orange dots called AHL. When these two binding together, it can further promote the synthesis of lysozyme and M-cherry. M-cherry is a kind of fluorescent protein. So when you use a fluorescent light, it can have this kind of red light emitted out. So when you see this redness, actually it can tell you how much amount of this lysozyme has been expressed. And when this expressed, it can repress the T7 RNA polymerase by binding to it. Once it binds to it, this one cannot promote the gene expressions. So originally, we designed these gene circuits trying to mimic the Turing patterns. However, we failed to do this because actually the Turing pattern needs all the things to be freely diffusible. However, T7 proteins are too big. It's stuck within the cells. So although we don't get the Turing patterns, we get this very, very cute ring core patterns. So then we want to understand under what condition these ring core patterns can be formed. Then we have this PDE model developed to describe the pattern forming circuit that I just described to you. We can see actually other than the ring core patterns, it has a capability to form all kinds of different patterns. So I call this no ring because it's only a peak, and this I call it one ring. So when you imagine it in 3D, it has a one ring. So based on this PDE models, if we want to know under what condition ring core patterns are formed, maybe that's very, very difficult. Because writing equations doesn't mean understand the mechanisms and how to find the key factor influencing the morphology of patterns. One way is to screen through the parameter space. However, if you have a simple calculations, this is a data set that we want to generate. And even if each of the data we need 5 minutes in numerical simulations, the total time is 3,470 days. Sometimes we can use the cluster to make it faster. However, even with the cluster, it still needs a long time to generate all these data. So then I propose this framework. If I can use a neural network to emulate the mechanism-based model, then if I use a neural network to screen through the parameter space, that will be much faster. So based on this idea, this is a framework based on the ideas. If I have a biological system, then I can develop a mechanism-based model. If by using numerical simulations to get the objective function or to investigate the system dynamics, it's very slow and sometimes impossible. However, if I use a very small scale data sets from the numerical simulations and use it to train a neural network, with a trained neural network, I can screen through the parameter space much faster. So on the right-hand side is the structure of my neural networks. The inputs means all the parameters of the systems. Then I have 500 LSTM cells. Each of the LSTM cells is associated with a prediction of 1 dot. So, yeah, sorry, I need to go back a little bit. So for this curve, because it's symmetric, actually I only need to train half of it. And then I discretize all these dots, this curve into 500 dots. Then for each of the LSTM, I can corresponding to one of the dots and do the predictions based on previous few LSTM cells. Then this is how my neural network works. This is I plotted the predicted data from the neural network versus the simulation data. And this is for distributional data, and this is for the peak value. And you can see they did pretty good jobs. And also, I selected a few from the test sample. And you can see all the predictions are correlated with the simulation results very well. And the trained neural network can emulate mechanism-based models with more than 30,000 times faster.
Shangying, if I can stop you for a second, with LSTM models, there's a high likelihood of them memorizing the training data, and especially with patterns, if they're symmetrical, they can easily memorize and reproduce the pattern. So I just want to know what you have done to alleviate that, or if you have seen something similar to that.
Oh, that's a very good question. So actually, when I do the training, I didn't use the symmetric data. I only use half of it. So it's first of all, it's not symmetric in the training data size. And secondly, all this data show in B, actually, the neural network has not seen these data. So that means actually, other than the training data sets, for the test data sets, it can also do a good job. And then furthermore, last but not least, on the next slide, this can perfectly explain your concerns. So this one is, so the top one, the top one is the one that I generated using neural networks. And this is the one that's existing in the neural networks. But all these are the ones that I found that had the 3-ring patterns, that has 3-ring patterns when I use neural network to screen through the parameters. So after I found all this, after I find all this, then I use the PDE simulations to validate whether these findings are true or not. And you can see for most of the cases, the neural network works very well. However, sometimes the neural network can see ghosts like this. Then, did I explain your questions?
Yeah, definitely. Thank you so much.
Yeah. So then I continue. Yeah, I've already talked half of these slides. For most of it, you can see the neural network seems to do very bad. Oh, excuse me for a minute. Yeah, sorry for that. Yeah, so even for a neural network that has accuracy about 99.9%, there's still 0.1% chances it will make mistakes. Then, how we know for each of our predictions whether it's accurate or whether it falls to that 0.1% not accurate groups? So, because we want to avoid this to happen, if for each of the predictions, we need to use PDE simulations to validate, then that compromised our proposal that we want to make it faster by using neural networks. So we developed this ensemble predictions. We want to know for each of the predictions whether we can, besides predicting the values, we will also want to know how confident the neural network is in predicting the values. So we use 4 neural networks.
Shangying, I can stop you for a second. So you mentioned that you're using an ensemble. Do you think by using, I just want to know how different these networks are, and also whether or not these networks can produce enough error in the system or variety in the system where you can use them to check whether or not your system produces valid results?
Oh, that's a very good question. Actually, for all these neural networks, the structure of the neural network is the same. The only difference is at the beginning, the random initialization process. And also, when we feed the data into the neural networks, we also random shuffle it. So the question that you have, maybe whether they have enough differences, I think definitely. First of all, because all these neural networks are very deep. So I didn't bring the curve here, but if you see the paper, the data set size I use is 10 to the fifth. But if you further increase the data size, actually the loss function can be further reduced. So that means the data size we choose, it didn't make the neural networks all converge to one global minimal. I suspect the neural networks may stuck in some of the local minimals, but that's very close to the global minimal. So most of the predictions the neural networks have are very consist. But for some prediction, if they totally disagree, that means they didn't do a very good job. Did I answer your question?
Yeah, thank you so much.
Yeah. So with these 4 neural networks, you can see there will be predictions. Then I'll choose the one predictions that has the least mean square error between all others. So I call this a voting process. So this framework not only can provide final predictions using voting, it can also give us an estimate how the neural network perform of each prediction. How we can do that, you can see on the right-hand side. So based on the disagreement in predictions and the error of final predictions, so maybe I need to explain a little bit. Disagreement in predictions, this term is calculated based on the mean square error between all these predictions. And this is error of final predictions, means the error of the final ensemble predictions with the ground truth. So we can see these two are actually correlated. So because when we do screen through the whole parameter space, actually we don't know the ground truth. So even though we don't know the error of final predictions for each of the prediction, we can estimate that based on our disagreement in prediction, because we can easily get this using the ensemble neural networks. If the disagreement in prediction is high, maybe the errors of final prediction are presumably high. So these are 3 examples. Based on this framework, now for each of the prediction, we not only can predict the curves, we can also predict the confidence score that the neural network performance is. Using that framework, then we'll do the real work. We screen through all the parameter space. These are all the parameters that the model have. Then we screen through all the parameter space to see under what condition, like how each of these distributed. Yeah, sorry, I didn't have the tag here. However, the orange one means, the orange dots means the 3 ring patterns. Then you can see the 3 ring patterns are formed somehow need some specific parameter range. like a very small KT and somehow big domain range and some others. So other than this, we also pick something that are very interesting, like this one and this one. So on the next slide, you can see if we fix all other parameters and only vary these two, then we can have an almost linear correlation between this parameter in order to generate the 3-ring patterns. And this is a curve fitting. You can see actually it's quite linear. Then with the other two parameters, we can see actually they're reverse correlated to each other. So because of the neural network, we're able to do all the manipulation of the parameters, and then we can get the objective function that we designed. Yeah, based on all this, actually, there are very interesting findings because actually, when I use this to screen through all the parameter space, I have a threshold. I randomly set a threshold that if it exceeds that threshold, I throw all the data away because I think because they have so large disagreement, maybe it's not reliable for me to trust its prediction. So I throw away some of the data when neural network do the screening. So that's what you can see. You can see for some of the space, all the data are gone. There's no data here. So that makes me think, oh, in this area, maybe neural network didn't learn very well. So if there's a way the neural network can give a feedback to the data generation process, maybe it can tell the data generation, hey dude, I need some more data in this area. So yeah, this is just an observation. So this can guide to my next research. If anyone is interested in discussion, I'm very happy to. So the key takeaway is first, neural network can emulate mechanism-based biological models. Then it can help with the circuit design in synthetic biology. And the key concept is to train a neural network using a limited number of simulations generated by mechanism-based models. This number is small, such that the simulation can be completed in a short time frame. It will not be that burdensome, but it also needs to be large enough to enable the reliable trainings. So you need to pick the data very carefully, because if you want a very, very big data sets, you want to achieve the global minimum, then it defeats the meaning of using these methods. Yeah. So third, disagreement in training is related to output accuracy. So an ensemble of neural network can be used to enable the safe content evaluation of the quality of each single predictions. Then I'll very briefly talk about my future plan based on the observation that I have. Because, sometimes even the small scale data that collected from numerical simulations can be very, very burdensome, because sometimes the simulations are very slow. Then I wanted to develop methods that neural network and the data collection can communicate, and the data connection feed the neural network some data, and neural network told the data connection at what area I need more data. So I think in a smarter way, maybe I can further reduce the data needed for this process and improve the data quality for the training. Yeah, that's all. Thank you so much for listening to my presentation. So I really want to thank my PI, Lin Chong Yo, as well as Catherine Heller, a professor in statistics department. So we do collaborations. Kai Fan is one of her students. And all this Nan and all this are from our lab. They help me both on experiments and also on developing these methods, as well as very meaningful discussions. And yeah, thank you. Also, thank you for hosting this. Yeah.
Thank you so much for a wonderful presentation. So I'm going to start with questions that were posted online. So one of the first questions that was asked is, did you use different initialization schemes for each member of the ensemble?
Yes. So for each of the ensemble neural networks, the initialization is random at the beginning. Yeah.
And was there any difference when you trained the models in parallel or separately?
I trained the model using the cluster. So they are in parallel, but they don't communicate with each other. So it can also be called separately. Yeah.
Okay. Another question that they're asking is that how would you imagine the process work? At latest, what are your initial thoughts?
Can you repeat that question? I didn't get it.
They're asking for future direction. How would you imagine the process work? Like for your future plan, you mentioned that you're going to provide feedback from. If you go to slide number 18,
Yeah.
I think their question is this.
I
So
see.
what you're describing is providing feedback from data collection to your neural network. They're asking how are you going to introduce this process?
So actually, I have a very preliminary results. It's also based on ensemble predictions. So with data collection feeding to neural networks, maybe at first only a very small batch, like 1,000. Then the neural network can estimate the sensitivity between the inputs and outputs. If there's some places in the neural network that did very bad, like the disagreement between the ensembles are very bad, then neural network can be able maybe to provide the feedbacks in this area, maybe around these parameter sets. I didn't do very well, so I need more data around in this area. Yeah, so I'm still wrapping up, so maybe my next paper will explain more on this.
Yeah, I think that answered the question. So I don't think we have any more questions online. I have some questions. One of the first questions I have is whether or not you used other recurrent neural networks for performing this. So, for instance, using GRU or using by-LSTM. Did you try any of those?
I only tried the normal recurrent neural network and didn't work. Then I found the LSTM, and then I tried it. It worked perfectly. So I didn't try other recurrent neural network. Maybe that works too. Yeah.
Okay. And if you go to the slide where you showed the biological mechanism that you're working on, and or maybe the simulation process that you came up with. Yeah. So just for better understanding of the audience, when you're using partial differential equations as a way to simulate the data, how much data or time is required for doing the modeling?
You mean the actual modeling or the simulation of the modeling?
The simulation compared to using deep learning.
So actually, after the model was developed, each of the simulation needs about an hour. But then after further investigating this model and do further simplification, so when we use this to do if we can simplify this, like we think it's symmetric, then we use a one-dimensional curve as output. Then it can be reduced to 5 minutes. Okay. So each of the predictions is 5 minutes, but each of the prediction from neural network is 0.01 seconds. So, yeah.
And just so that the audience understands, what are the outputs from your simulation or the inputs to your deep learning model?
So the output M cherry is corresponding to the lysosome. So in the experiments, when you show the M cherry, the patterns, actually it reflects how much the lysosome has in the space. So this is the L here. Yeah.
Okay, that's great. Let me just check if there are any other questions. It seems like we don't have any other questions. With that, I would like to thank you so much for giving this wonderful presentation and accepting our invitation. And I would like to thank the audience for joining us today. Stay safe, and thank you all.
Thank you for hosting this. Yeah, I really enjoy it.